Good day, everyone. How are you today? Hope we're all doing great. Welcome to this Google Classroom Genetics. I am Diana T. Duma, BSED Science 3A student, and for today's video lesson, we're going to tackle the DNA structure and replication. So prepare yourself together with your ball pen and paper for this another learning experience. But before we start, let's say with a short prayer.
Our loving and generous God, we praise and thank you for the bountiful blessings you have given us. Thank you for giving us a mind that can know and a heart that can love. Thank you for giving us the chance to continue learning amidst the pandemic that have caused a lot of changes in our lives. We are sorry for the times we have failed you. We humbly ask for your forgiveness. Father, help us to focus on our studies. When learning becomes difficult, grant us the gifts of courage and enthusiasm. Grant us the courage to use our knowledge in making a difference into the lives of the people around us. All this we pray in your mighty name. Amen.
But before we begin, please be reminded our 3 classroom rules. What is the first rule, Junami? Okay, very good. Raise your hand when answering. What is the second rule? Kim? Okay, very good. Listen to your teacher during the discussion. And lastly, what is the third rule? Isis? Okay, very good. Respect each other.
Before we start our discussion, let's have an activity first, entitled 4 Pigs in 1 Word. All you have to do is to guess the scrambled letters, and each item I will give you 10 seconds to answer it. Based on our activity, what do you think our lesson for today? Okay, very good. Our lesson for today is to describe the DNA structure and to explain the DNA replication process. Before we start our lesson, can you still remember our previous discussion? Okay, very good. Our previous discussion is all about sex chromosomes, wherein XX chromosome stands for female and XY chromosome stands for male. Before we start our lesson, get 1/4 sheet of paper, and I have here sets of questions that you need to answer. And I'll give you 10 seconds to accomplish each item.
At this point, we will be having a new activity entitled Find My Pair. I will group the class into two. From Alcuetas to Donghit, you will be the group 1. And from Duma to Toscano, you will be the group 2. For each group, I will give you a sample of DNA sequence, and what you will going to do is to find the nitrogenous base pair. Is that clear? Again, is that clear? Okay, very good. Your time start now. And now, Group 1, present your activity to the class. Okay, let's give a hand to Group 1. Next, Group 2, present your activity. Okay, very good. Let's give a hand to Group 2. But before we proceed to our discussions, who among you here has an idea of what is the DNA? Okay, Mark. Very good. DNA stands for deoxyribonucleic acid. Who else? Okay, Jonami. DNA is? Okay, very good. DNA encodes all the information of all living things. Okay, your answers are all correct. The DNA encodes the information that cell uses to synthesize a protein. The DNA can also replicate by passing its information. The DNA also contains the genetic information of all the living things here on Earth. So, who do you think discovered the structure of DNA? Anyone? Okay, since you don't have any idea, in 1909, Russian-American biochemist Phoebus Levin identified 5 carbon sugars, the ribose, as part of some nucleic acids. And in 1929, he discovered a similar sugar, the deoxyribose, and other nucleic acid. He had revealed a major chemical distinction between RNA and DNA. RNA has ribose and DNA has deoxyribose. Levin also discovered the 3 parts of a nucleic acid: a sugar, a nitrogen-containing base, and a phosphorus-containing component. He deduced that a nucleic acid building block must contain one of each component. Another person who discovered the structure of DNA is Erwin Chargaff. In the early 1950s, 2 lines of experimental evidence converged to provide direct clues that finally revealed the DNA structure. An Australian-American biochemist Erin Charga showed that DNA in several species have the same amount of the bases. The adenine and thymine has the equal amount of the bases guanine and the cytosine. Class, can you still follow? Okay, very good. Next is the English physicist Maurice Wilkins and English chemist Rosalind Franklin. They bombarded the DNA with X-rays using a technique called the X-rays diffraction. Then, they deduced the overall structure of the molecule from the pattern in which the X-rays were deflected. Now, let's proceed to the structure of the DNA. What is a gene anyway? Okay, since you don't have any idea, a gene is a section in DNA molecules whose sequence of building blocks specifies the sequence of amino acids in a particular protein. The structure of DNA is easiest to understand if we begin with the smallest component. A single building block of DNA is called nucleotide. Again, class, what do you call to the single building block of DNA? Okay, Jomark. Very good. It's called nucleotide. It consists of one deoxyribose sugar, one phosphate group, and one nitrogenous base. So here is the illustration. The blue parts indicate the nitrogenous base, the green part indicates the deoxyribose sugar, and the yellow part indicates the phosphate group. Those three are the parts in a single nucleotide. Is that clear? Okay, very good. Okay, so what are the four nitrogenous bases in a DNA? Anyone? Okay, Chancille. Very good. The four nitrogenous bases in DNA are the adenine, guanine, cytosine, and thymine. Okay, so among the four nitrogenous bases, what pairs is considered as purines? What about pyrimidines? Okay, adenine and guanine are purines, while cytosine and thymine are pyrimidines. What do you think is the difference between purines and pyrimidines? Okay, you don't have any idea. Adenine and guanine are considered as purines because they have two ring structure, while cytosine and thymine is considered as pyrimidines because they have only one ring structure. Is that clear? Okay, very good. What do you call to the single building block of DNA? Okay, Erica. Very good. Nucleotide. Nucleotide joins into long chains when chemical bond form between the deoxyribose sugar and the phosphate. They create a continuous sugar-phosphate backbone. Here is the illustration. The green part and the yellow part are the sugar-phosphate backbone. Can you see it? Okay, very good. And now, try to observe this illustration. Okay, Marcel, what did you observe to the illustration? Okay, so the DNA structure is arranged in inverse manner. What about you, Maricar? Okay, very good. They are arranged in opposite direction. Okay, so your observations and ideas are all correct. The opposing orientation of the two nucleotide chains in DNA molecules is called the antiparallelism. So what is antiparallelism? Okay, you don't have idea. Antiparallelism becomes evident when the carbon of the sugars are assigned numbers to indicate their position in the molecule. So here is the illustration. Based on the illustration, each side of the sugar has number from 1 to 5. Can you see it? Okay, very good. In short, the orientation or the position of the sugar what makes the DNA structure anti-parallel. Is that clear? Okay. Next, DNA is called directional. Antiparallelism in a DNA molecule arises from the orientation of the deoxyribose sugar. One half of the double helix runs to 5 to 3 direction, and the other half runs to 3 to 5 direction. It is pronounced as 5 prime to 3 prime or 3 prime to 5 prime. Is that clear? Okay, very good. Here is the illustration. As you can see, from the top, it is numbered from 5 to 3, while the other one, it is numbered from 3 to 5. Okay? Okay. The symmetrical DNA double helix form when the nucleotide containing adenine pairs with those containing thymine, and nucleotides containing guanine pair with those carrying cytosine, because purines have 2 rings and pyrimidines has only 1. These specific purine-pyrimidine couples are called complementary base pair. Again, class, what do you call to the purine-pyrimidine couples? It is the complementary base pair. Now, let's discuss about DNA replication. But before that, class, what do you think is the importance of DNA replication? Okay, Zacarias. Very good. To make a copy. What about you, Quirel? Okay, very good also. To pass the genetic information. Okay, class. What is DNA replication? Maricel? Okay, very good. DNA replication is to make a copy of a new DNA. So, DNA replication is the process by which a double-stranded DNA molecule is copied to produce 2 identical DNA molecules. Here is the illustration. Can you see it? Okay, very good. And now, why is it DNA replication said to be semi-conservative? Okay, you don't have idea. DNA replication is said to be semi-conservative because each new DNA double helix conserves half of the original. Again, it is considered as semi-conservative because each new DNA double helix conserves half of the original. Is that clear? Okay, very good. Anyway, class, there are 3 hypotheses to DNA replication. The first one is it is semi-conservative. The other one is conservative. And the last one is dispersive. In semi-conservative replication, it states that the half of the DNA is being conserved. While in conservative replication, from the word itself, it conserves all the strand of the DNA. While in dispersive replication, the genetic information is being scattered or distributed. Here is the illustration. Can you distinguish the difference between them? Okay, very good, class. And now, let's discuss about the steps in DNA replication. The DNA replication occurs during S phase of the cell cycle. Again, class, on what phase of the cell cycle does DNA replication takes place? Okay, very good. It is in the S phase. When DNA replicates, it unwinds, breaks, and builds a new nucleotide chain and mends. Enzyme called helicase unwinds and holds apart replicating DNA, enabling other enzymes to guide and assembly a new DNA strand. Human DNA replicates about 50 bases per second. A site where DNA is locally open, resembling a fork, is called replication fork. Again, class, what is replication fork? Okay, very good. It is the site where the DNA locally open and resembling a fork. The illustration shows the enzymes in DNA replication. First is the helicase. Next is the binding protein. Next is the primase. Next is the DNA polymerase or DNAP. And last is the ligase. The DNA replication begins when an enzyme helicase breaks the hydrogen bond that connect a base pair. The binding proteins holds the 2 strands apart, then another enzyme primase then attract complementary RNA nucleotides. to build a short piece of RNA called the RNA primer at the start of the segment of the DNA to be replicated. The RNA primer is required because the major replication enzymes, DNA polymerase or DNAP, can only add bases to an existing nucleic acid strand. A polymerase is an enzyme that builds a polymer, which is a chain of chemical building blocks. Next, the RNA primer attracts DNAP, which brings in DNA nucleotide complementary to the exposed bases of the parental strand. This strand serves as mold or template. The new bases are added one at a time starting at the RNA primer. Next, the DNAP work directionally, adding new nucleotide to the exposed 3' end of the sugar in the growing strand. Overall replication proceeds in 5' to 3' direction because this is the only chemical configuration in which DNAP can add bases. Next, an enzyme called ligase then seals the sugar phosphate backbone of the pieces, building the new strand of the DNA. The DNA polymerase also proofreads as it goes, excising mismatched bases and inserting correct ones. It is also removes the RNA primer and replaces it with the correct DNA bases. Yet, another enzyme called annealing helicase rewind any section of the DNA molecules that remain unwound. Finally, ligases seal the entire sugar phosphate backbone, and the ligase come from a Latin word meaning to tie. So, that's all for the DNA replication. Do you have any question? Are you sure? Okay, since you don't have any question, get one for sheet of paper and I will post your quiz on our Google Classroom. Is that clear? Okay, very good. Okay, class, for your assignment, study the structure of RNA. So, do you have any question? Clarification. Did you learn something today? Okay, very good. So that's all for today. Goodbye, class.